Very good evening, everyone, and welcome to Saliva Annex. I give a hearty welcome to all of you in today's session. Before that, let me introduce myself to you all. Well, I am Dr. Gargi Roy Goswami, an interdisciplinary oral healthcare professional, a geneticist, and a general dentist by profession, an academician by passion, and a scientist by heart. So, with that small introduction, let me just go into today's session, and I hope all of you will enjoy this session with me. And also, I would like to thank everyone who has joined today because uh, we have participants. We got a huge response, and thank you for that. And we have participants from all over the world, from Brazil, from Spain, Portugal, Egypt, and many more places. And thanks to everyone for joining. Well, I will not waste any more time and let me just share my screen with you. Give me 2 minutes, please. Okay, so I hope you all can see it. And here I go. Sometimes it takes little time. Yeah, so that is it. Well, with an ambition and a mission and a vision to create, collaborate, and contribute to science, today I am Dr. Gargi Roy Goswami once again in the platform of Indian Academy for Clinical and Dental Genetics with you all uh, into a topic that we will discuss. But before that, I would like to give my prayers and gratitude to my father, Dr. Kamalesh Chandra Roy, because whatever today I am doing and whatever I am is just because of my dad, and he is my hero. He was the first dentist of Murshidabad district. That's a district town in West Bengal, India, where I belong from. Well, uh, let's go into the first part of our session today. So, we were supposed to talk and try to understand in the beginning, like how can saliva serve as a tool for oral cancer diagnosis? Well, because today we are talking about saliva diagnostics and oral cancer diagnosis and its typical role in it. And if you have gone through the what you will learn from the webinar topics, I'll just go accordingly. Okay. So, how can saliva serve as a tool for oral cancer diagnosis? Let's see. We will just try to find out the answer. Okay, so this is my favorite one, and as you all know, who have been following my sessions, and all those who are new here, for everyone, let me tell you that I like to describe saliva just by quoting Dr. Irwin Mandel, as in, saliva lacks the drama of blood, the sincerity of sweat, and the emotional appeal of tears. How wonderfully it is described, and that's the way to describe saliva, I must say. Well, basically, what is saliva? It is a watery, slightly alkaline fluid which is secreted into our mouth and through the salivary glands and mucous membrane, that is the membrane that lines our mouth. Basically, it's a watery and frothy kind of liquid, okay, that is being secreted by the salivary glands, and it is made up of many substances. So it's kind of a highly complex mixture of substances. So we will see whether those substances can help us in finding out oral cancer or not. So let me proceed to the next slide. Many of us might know that how saliva is generated. So this is just basics. We'll just turn on the basics for all of who are attending. So basically, saliva is generated by particular cells in the salivary glands that are acinar cells. And what happens is they get collected into ducts, and finally they get released through the opening of the duct that opens inside our mouth in the oral cavity. So it's just like basically you have a tank to store water, and then when you want the water to come either into the kitchen or into the bathroom, so when you open up the tap, what happens is it flows through that pipelines and comes to that particular bathroom or kitchen tap, right? So that is how exactly the mechanism how saliva is generated through these salivary glands, and it flows down right into our mouth through the particularly major salivary glands that we have that constitutes 90% of the saliva that is secreted. Namely, they are parotid gland, as you can see in this photo where I hope you can see my cursor. So there, that is where I run my cursor. And then there is submandibular gland and sublingual gland. Apart from that, we also have minor salivary glands, like it's present in the lips, the labial, depending on the position, where they're present. So that is how they are named. So they also definitely contribute to some portion of the salivary secretion. Okay. Now, before we go and understand that how saliva can be help us to diagnose oral cancer, for that we need to understand why is saliva so important, right? Okay, just putting it in a very simple way. Saliva is important because, as I said, it's just our spit, right? So it's a fluid that is produced by the salivary glands, and it is rich in many proteins and many other biochemicals that are found in blood, as well as if it gets filtered into the mouth. Now, these proteins or biochemical substances can be normal ones, or it can be related to certain diseases, like it can be cancer, it can be viral diseases like HIV, and so on and so forth. I mean, there are a huge number of examples. So, to become a promising diagnostic fluid, what happens if, like, there are some physiological levels for any biochemical moiety or any molecule that way, if I put, like even in blood. So same way in saliva. So it gives us a picture about the physiological, as well as if with time any pathological conversions happens. So what happens is certain biochemicals or certain molecules may increase or decrease accordingly, and that is how saliva is so important and becomes a very promising diagnostic tool. Let's move on. Yeah, I know that many of us know this. Saliva is basically defined as body's mirror. It actually resembles whatever is there in the blood. Yes, that's very, very true. And I will just justify this with my later slides. Hang on. So, if you see the normal composition of the saliva, like as in you see here, it has enzymes, it has antimicrobial proteins, immunoglobulins, ions. Uh, I don't know to explain so much. I mean, there are so many things. I think if we just start explaining one by one these components and functions of saliva, I think we can actually talk hours together. So this is one part. So that means these all can be potential substances that might be related to certain diseases. So as I was telling that some enzymes can go down or some can come up, or it may be certain, like in infectious diseases, it can be the, up and down of the immunoglobulins. And example, I'm just giving few examples. So that means I can just measure all these normal things in saliva, as well as if something goes wrong and the cells becomes cancerous. So something of the cancerous cells, whether it is the DNA of the cancer cell or any other things, or the cellular debris or any other vesicles, we can get that even into the saliva, because it comes from the blood to the saliva, as well as definitely it is present in blood. So it comes to the saliva also. I'll come to that and we'll understand how. And that means also we can have an idea of different types of maybe inflammatory cells, maybe some soluble factors, or I mean the whole tumor microenvironment. There will be so many changes, right? So we can actually try to assess all those in saliva. We can get them in the saliva, just because whether it is normal components or whether it is changed components that we were just talking about, it happens because biomolecules from blood to saliva, they use normal cellular transport mechanisms like diffusion, active transport, ultrafiltration, translation. So it differs from case to case, from biomolecule to biomolecule. So that's why saliva is called as body's mirror, because whatever there is in the blood, it is most of the things are present in saliva. I am not talking about any quantity or quality, but yes, that's why saliva is very, very promising and very, very well known as the body's mirror. So I hope I could justify that statement. Let me go forward. So that means if I want to use saliva as a diagnostic fluid, okay, so it can be, I can choose anything. So that anything can be either DNA, it can be antibodies, it can be lipids, it can be proteins, it can be metabolites, microbes also, RNA obviously, and many more things it can be. All right. So that means whatever I'm trying to diagnose, that thing should be, that molecule should be traceable. It should be a traceable substance that exists in the saliva, and that can help to detect or even predict certain diseases. So that's as simple as that. So that means, what I mean to say, the normal, abnormal, whatever components can serve as what we generally call as salivary biomarkers. That's very fascinating. Now, with the biomarkers, I can actually diagnose a disease. See, this is not only for oral cancer because saliva has immense applications. Maybe I can speak on that in a different webinar, just in which areas saliva can be used. So, basically it can help us with all the salivary biomarkers. We can definitely go for a disease detection, we can try for risk assessment, we can diagnose the disease, we can, that can also help us in prognosis. Not only that, we can also venture into monitoring of a disease or response to treatment of a particular individual or group of individuals. So that's powerful tool, or that's the power of salivary biomarkers. So what is this saliva diagnostics? So let me just put it in a very straight and simple way. Saliva diagnostic is basically the study of your spit or saliva in a better way. So what scientists do is they try to look for certain elements or compounds or biomolecules, however you tell them, and that can help them to diagnose and predict certain diseases. As simple as that. So here we are talking about oral cancer, right? So let's see what we have in the bag. Well, now coming to the second part is how should saliva be collected for oral cancer testing? Now, before that, I'll just like to add on that, as you saw that we have a very complex mixture of substances in saliva, and as well as it's in our mouth. So in our mouth, it's not only the secretion that comes from this saliva ducts, but also we have food debris, then sometimes we have something called as gingival clavicular fluid, and we might have some infections due to those microbes will be there. And anyway, we have commensals that we all know, and we might have some other pathologies which might contribute even to the composition, changing the composition, and definitely other factors. So all this together, what we call is known as whole saliva. It includes everything, even the food debris particle when considered, which is present, which most of the time is present. So that is whole saliva. So there are certain ways that we collect saliva. Yes, many of us are aware. The most traditional and conventional, and rather sometimes taken as a gold standard, is to spit in saliva, or which we generally technically we call it as passive drool technique. But yeah, so that's case to case, it varies. So let me just dive in and give you a tour through certain collection tools that will really enrich today's session, and you will understand how exactly this collection procedures, affect your downstream experimentation or whatever analysis you want to do. Because, as I was saying, that we can work with DNA, we can collect RNA, we can collect antibodies, we can collect proteins, and etc. etc. So, depending on what molecule we are focusing on, definitely our collection strategy will also change. Just giving you a short tour. Yeah, but before that, I cannot miss this slide. I cannot miss this slide because when I'm talking about saliva and I'm trying to justify saliva against blood, yes, so definitely it has many advantages, and maybe many of us know about it. First of all, the collection is undemanding. So if you ask saliva from someone, at least in India, nobody will ask you what you will do with that saliva. Yeah, so, but for blood, it's always scary for the person. Also, and definitely, collection of saliva is non-invasive. So there is no scalpel, no syringe, no prick, nothing. So that makes patients to be very compliant, particularly patients who are children, who are geriatric patients, or those who are medically compromised, or with any other special care situation. So also very important is that there is no need of any trained medical staff. Like we need a phlebotomist for taking up the blood, but for saliva, I think if I just teach anyone, and it can be self collection, even the patient can do it by themselves. And also, multiple stem cells can be collected very easily, and saliva samples are quite safe to handle. There are minuses, but I'm not going to tell you that right now because I'm telling the advantages of saliva. Well, and comparatively, comparing with blood, samples are easier to ship and store. And the very important part is that there are minimal risks of cross contamination, which is very much present with blood sample or any other biological sample, biological fluid sample that way. Okay, so I was talking about certain devices. So this is one device with which we when we talk about passive drool, generally we what we do is we take up some vial and we tell the patient to spit in, but that's not the correct way. There are methods to do it in a conventional way, but these are certain collection devices where it guides the patient, and if you see, I'll just move my cursor. I hope you see that. So these are all these graduations made. So that means you can actually see how much volume of saliva is being collected. Okay. So, and also this funnel kind of thing will help the saliva not to splatter around, or you won't lose the sample actually. So it's very easy for the patient to just give you sample through passive drooling using this particular Simply O5 saliva collection device. Now, if I want to work with salivary DNA, definitely the scenario changes. So earlier, which I was talking about, it's for whole saliva or when I need a large volume of saliva to work on. And now when I want DNA, so definitely I will collect it from the saliva and also from the cells, in the buccal cells, right? The inner part of the chick. So if you see, there's something called as DNA cell. If you see, this is a box, where there is a solution. Now, this solution in most of the commercial devices, this solution is present, which is called as stabilizing solution. Well, the composition is proprietary to the companies. So basically, what is the function? The function is to keep your molecule of interest intact. Like, for example, here we are talking about collecting DNA. So with this, you can keep DNA at room temperature, like around 37 degrees centigrade, even for a month. So it's very easy to transport your DNA with not degrade, because DNA is real. It's a real tough guy. It doesn't actually give you pain like RNA and protein. All right. So another device is called a supercell. With this is a fantastic device where it's just like a cotton collection pad. And if you want to collect again whole saliva, or you don't want, you want to collect a protein like enzymatic acids, or these are very useful for patients who have less saliva flow, or maybe children, like teenagers, adolescents, okay, and not only that, for everyone, because it is very compliant. They just have to dip it down below the tongue, and what happens, this cotton absorbent pad slowly collects, and there is an indicator which tells, okay, that's all, I don't need more saliva. So this is another interesting one with which you can, when you think of working on say salivary enzymes, hormones, or any other, rather I can tell this particular device is kind of a universal universal collector. So it has many applications. All right. And if you really want to do a very high-end, like techniques, like you want to go into proteomics or transcriptomics kind of analysis, or maybe some high-end technologies where you really need a very, very pure sample. So this is another very fantastic device. And the best part is, this is just like the one you saw before, like SuperSal. But the thing is, here there is a special filtration unit, ultrafiltration unit. So at the first go itself, once you the same way you collect by the absorbing, I mean, by the absorbent pad of this particular device, and then you push this saliva sample through this filtration unit. So what you get is, you get two separate samples in two weapon drops. One will have salivary proteins, one will have salivary RNA. How wonderful is that? And there will be no cell debris, no mucin. You don't want DNA, so there's no DNA because you are working with RNA, and you want to work with protein. So right at the collection level, you can do the separation. You can collect a very, very purified sample. So it's a wonderful device. So these are the ways, and particularly when we talk in perspective of oral cancer, it can be DNA. So we have DNA cell. It can be cell-free DNA, or it can be exosomes. So there are many things which I will talk about. These collection devices would be very, very helpful. Well, in this respect, I want to tell you one thing, that I have started an online Salivanomics workshop program. So that's a 3 days workshop, and we had a very jam-packed session in a few days back in May. So we are coming back again in June. So please mark your calendars for 20, 21, and 22nd June. And this is done entirely with live sessions. And here, if you want to know more into saliva, the practical applications, the troubleshooting of working with saliva, and many more very interesting stuff. So go and visit www.geneden.com, and there you will get all the details. Well, let me come back to our presentation, and now I will tell you certain popular molecular technologies for oral cancer diagnosis. Okay, so before that, we all know that oral cancer can look like, clinically, it can look like as simple as a small patch like this, or something dreadful like this on the right hand side. So that means this is a classical picture that we have been very, very familiar. So it can be that one cell began certain changes, and then those cells kept on multiplying, and they kept on occurring more changes, and then from a precancerous stage, it moved to something called as invasive cancer, or what we call as metastasizing cancer, right, or metastases. So this is a typical epithelial dysplasia that is seen when we see oral cancer tissue, maybe at different stages under the microscope. Well, I'm using very, very common terms because it's a very versatile crowd. Our audience is always like that. Okay, so basically what happens, let me go dive deep down a little bit more. So maybe one cell gets a mutation in the DNA, and then those mutated cells proliferate. And, so one bad guy gives rise to a group of bad guys. And then in that, again, more mutation or epigenetic changes happen. That again proliferates. And finally, it goes to tumor progression. Slowly, more once the cell becomes, DNA starts getting mutations due to many reasons. So one bad guy will pull in the entire colony together, and each one of them becomes different domes. As you can see, they're shapeless. You cannot predict them. So that is how exactly the genetic mechanism that happens. There are many things inside. So why I told you that, because based on these, there are different technologies, molecular technologies, with which we can pinpointedly identify, or we can differentiate from a normal cell and a cancer cell. So one of those fantastic molecular technology that I must tell today is microarray technology. So microarray technology is basically, it's kind of a small glass or nylon membrane platform where you have, very small microscopic grids. And the best part is you can analyze DNA, RNA, or protein with any of the thing, and in a few time you can actually analyze hundreds and thousands of genes, different genes together, and you can understand. Also, you can differentiate between normal cells and oral cancer cells or any other cancer cells. So that's a fantastic technology. Another is proteomic analysis. So this is basically where your sample has proteins, and then you separate them by a process called electrophoresis, where we pass the solution, protein solution, through a gel-like structure, and it gets separated into bands of proteins where according to the molecular weight. All right. So in this, you can understand exactly the entire protein profile. That means if you have collected a saliva sample or a blood sample, see, these molecular technologies are not specific only for saliva, but can be used for blood. It is being used for blood. So now we talk about saliva. So because from saliva, if we get proteins, and we can understand what different kinds of proteins are, present. So that is how we, just separate the proteins, understand its differences, fragment them, and then we do some analysis, and we have the data to understand. So basically, this particular analysis provides an opportunity to investigate factors that are involved in the disruption of cellular structure, because that is definitely there, and also the function that leads to a cancerous stage or any other disease state. Now, I was talking about a little bit genetics, right? Mutation and stuff. So there can be different types. There can be pinpointed, like deletions. There can be extra nucleotides coming in. There can be inversion. There can be something called as translocation, like one part of a different gene comes and sits on the head of the other, and it becomes a totally mess. So a very fantastic technology with which we can, there are many things in this, so I've just put it up in a very simple way. I have tried that. Well, so what happens is, even if you want to find out this kind of, whether one nucleotide has got deleted, whether there is an extra one, or what exactly has gone wrong, then definitely there is something called as sequencing, and we are in the third generation, and that's called as next generation sequencing. So here, what we do is we can analyze like different human genomes or different human DNA. Like say, I take my DNA, I take yours, and whoever is present, and I take from someone who are from different countries, regions, and I can analyze it in one go. And basically, it is just cut. If you see this figure on the left hand side, it is cutting the DNA, labeling them, because DNA is a very big molecule, and then just chopping it off in different sections, and then labeling it so that we can reorganize it and understand. So there are different types again, but well, you can actually see the entire genome, the number of genes that has gone wrong or that are present. That is called whole genome sequencing. Or you can just focus on only protein producing genes, like that's called exome sequencing, and there are many other things. So that's again a very fantastic molecular technology for used much, and it is being very much used in diagnosis of oral cancer. It gives you these technologies, these genetic technologies or molecular technologies, gives you pinpointed answers of whether chemotherapy will work or not, whether like surgery will work or not, whether there should be a combination of radiotherapy, chemotherapy, stuff like that. It's amazing. It's fantastic. Okay, so with this thing, I would like to announce very gladly to all of you that we save the date of 3rd July because our academy, Indian Academy for Clinical and Dental Genetics, is coming up with a fascinating online course on oral cancer genetics. So this is basically a refresher course from where we will take you right from basics to advanced concepts and don't worry and don't think that this is again a textbook stuff which will be taught. No, but yes, definitely we have to include the basic things. That's why it's a basics and how you can apply. You will understand the different technologies a bit that I was talking about right now, and how these are actually applied and how you can you know come up with new technologies or new ideas using these technologies, and definitely how more deeply saliva diagnostics is used, because that is one aspect which has to go, we have to take this thing forward, because saliva is a wonderful biological fluid, and that's why we are talking today. So if you want to see more about this program, go to the website www.genedent.com, go to homepage. There will be a tab called courses. Click that. Then you have certificate course. Click that. You will have a general idea about what we will be covering in this particular online certificate course of oral cancer genetics. I am very excited, and I hope you will be joining me in this course definitely. Okay, so coming to the concluding part, I will tell you now fantastic advances or recent advancements in a global perspective that has happened with saliva in the field of oral cancer. So there are emerging applications of saliva with something called as liquid biopsy, exosomes. All these can be collected from saliva, and they are used, very highly used, in oral cancer research, in oral cancer diagnostics. So, and also cell-free DNA and point of care. So point of care is basically what we mean by chair side, or some stuff or tests that we can actually do not, for that we don't need a very very grand laboratory setup. So just giving you snapshots of what is liquid biopsy. Basically with this particular thing we can use blood or saliva, and particularly saliva we can use, and there we can actually detect circulating tumor cells. That is what is meant by CTCs. We can detect exosomes and cell-free DNA and cell-free RNA, etc., etc. Because all these are basically products due to, as I was talking about, these mutations and all. So these kind of things will keep circulating in the body fluids of cancer patients, right? Because all these are happening because of these oncogenic mutations. So in case with saliva, it's very, very promising field where saliva diagnostics is applied in the diagnosis of oral cancer. Well, going to exosomes, see, these are very small microvesicles, I can see in the picture, and they are secreted by variety of cells. And the best part is, what happens is, exosomes are actually membranous structures, okay? They are nano-sized. This kind of, you have heard about nanomaterial, so their diameter is in nanometer. So these vesicles, they are released by many cell types. So if anything is going wrong, you can isolate exosomes from saliva in a very simple procedure. There are very simple technologies to isolate exosomes, because these exosomes will carry nucleic acids or proteins. So that means these, if it is cancer cell or normal cell, I'm talking about oral cancer cell particularly. So I can easily identify and pinpointedly understand and analyze with exosomes, salivary exosomes. That's the fascinating area. And cell-free DNA is nothing but, when these cancer cells are, just disintegrating, because cells will die, right? So if this is a, say, for example, this blue one is like a circulating tumor DNA. So that is called as cell-free DNA. Even this you can isolate cell-free DNA very easily from saliva using specific collection tools also, obviously. And I will focus on this test. Well, this is not available in India, so maybe some of us should one day develop, and very soon we should develop. So Salimark is OSCC. This is specifically for diagnosing oral squamous cell carcinoma. This is available with a certain company called as Periorix. So this is the only test that is available from saliva for OSCC. Well, it does not give initial or early detection, but it definitely tells whether it goes it's going bad, or if it is not so good. Then another one, it comes to the point of care device. This is a very, very fascinating device which works with a technology called as light box image analysis module, that is LIAM, and there's the same technology that is used in our smartphone. How wonderful, right? And this particular device is very fascinating. Now, presently, you can actually do qualitative as well as quantitative assays with this. Presently, it is standardized for for measuring salivary cortisol. Okay, but this machine is so potent that if you have done some experiment and you have some results, and obviously from saliva, and you want to standardize them as a test with this machine, you can do that. So that is another very wonderful thing. And if you want to know about this, you can contact me anytime. Okay, so another one which is very fascinating and very interesting is Efore. So that is electric field induced release and measurement. So the wonderful technology with this particular technology, what they can do is they can directly identify EGFR mutation. So what is EGFR? Epithelial growth factor release. All right. So EGFR mutations can be directly analyzed in saliva. Particularly, this technology is now getting upgraded to oral cancer, but it's in the development. Very shortly, it will be done. But it is now validated for lung cancer. Okay. So that's what that was the farm, and these are the fascinating advances that are happening. There are many more things which I couldn't put up because there is a limitation to this webinar. Otherwise, I will speak up to 12 in the night. All right. So once again, reminding you about the workshop in June and 3rd July when our 2 months online certification course starts. Please do join in and mark your dates. Well, I will definitely request you all to subscribe this channel of mine. And if you like this video, because Saliva Annex is something that is a free webinar series that I do almost every month. I try to be very regular, but sometimes I do not get time. But yes, I am trying to do it every month. So I will see you again with some other interesting topic related to saliva diagnostic next month. And I have something interesting to tell you. In the same channel, if you just type Eclectica, well, that's an interview show which I host. And in this, what I do, I call people from different areas of science, technology, then, healthcare, then entrepreneurial stories, inspirational life stories, photography, fashion, finance, many things. It's like multifaceted show just for keeping ourselves motivated. It's an absolutely voluntary endeavor. So we have done some 7, 8 episodes. So if you are interested, whenever you're free and you want to know something new, just go and watch the episodes of Eclectica. Okay, follow us in Instagram and LinkedIn. Follow me in LinkedIn, please. And also, we have our Croynas Facebook page. And please join our Facebook group of Indian Academy for Clinical and Dental Genetics, because there we do certain very, very interesting stuff. We do some live discussions at every Wednesday that we call as Let's Discuss Wednesdays, and we have showcase Sundays in that we pick up any member or any member of the group if they want to come up to showcase themselves, showcase their talent, or tell about themselves, about their work, about their achievements, and or any other thing that they want to speak out to the world. So we host them there. So, well, with that, yeah, I must thank all of them. This is my beautiful mom, and I always thank her for bringing me to this wonderful world. And this is my handsome husband, Debraj, and my very good friend and my backbone support. And these are the girls who lives my passion. That is Dr. Shruti, Miss Kanchan, and the very gorgeous Dr. Udita. So they are just like my wings, I should say. So, with that, thank you so much for being here today. And I think you have enjoyed these sessions. And if you have any questions, please write in the comment section, not in the live chat, but in the comment after the video, because then I will answer them offline because I think it's quite a time and everybody is hungry because in India, the it's dinner time. So I think, okay, so that was wonderful. And I hope all of you enjoyed this. And I will come back again with another episode of Saliva Annex. And till then, be safe, stay calm, and take care. Thank you so much. Bye bye and good night.